All right. Next up on the agenda is a discussion panel formatted similarly to what you saw yesterday. We are going to invite a handful of scientists from Just-Evotec Biologics to join us to talk about the rollout, adoption, and results of implementing the Biologics ELN that I had discussed earlier. So let me start with some brief introductions, and I'll let our panelists add to that if they have interest in doing so. We are joined by Tara Kulas, who is an analytical scientist at Just-Evotec, and I would say that she had spearheaded much of the organizational effort to find an ELN that was of interest. We are also joined by Abigail Brownell, who's a scientist in process design and part of Just's small bioreactor group, and Abby Neumann, who's a scientist in Just's media development organization. Hello, everyone.
Hello. Hi.
Okay, so brief relationship introduction here. You, of course, already heard me talk at length about the Biologics ELN, but I wanted you to hear firsthand about the experience of working with us on a fairly major project like this. Let me briefly tell you about the history of our relationship with Just-Evotec Bio, excuse me, so that we're all sort of starting on the same page. So Just-Evotec Biologics is an integrated design company focused on technologies to accelerate development of biotherapeutics, as well as substantially reducing their cost and time. And they have the explicit mission to apply those technologies to dramatically expand global access to biotherapeutics. Now, we have been working with Just Bio since the inception of LabKey Biologics. They helped shape the product and continue to help us refine and improving it. We work very closely with them, including the wonderful scientists on the panel today, and invite all of our partners to do so as well. So as Tara conveyed last year, for those who had the privilege of hearing her then, we built this ELN with this group of scientists and others to try to round out the scientists' experience when they were using LabKey Biologics and further centralize their operations and data. We just didn't think that documentation needed to be so painful or disconnected from a scientist's other contribution to their field and institution. So we worked really closely with Tara, Abigail, and their team of sleuths to build something that wouldn't just reduce the burden, but actually be desirable. That played out as some deep requirements gathering, looking across their institution and others, many design iterations with a lot of feedback sessions, an initial release for the ELN sleuths at Just for testing and feedback, and then a rollout earlier this year. My illustrious panel, first of all, in general, can you talk about anything you'd like to add about your role at Just, as well as what your role was in the process of rollout and adoption at Just Bio?
Well, as Bernie said, I'm an analytical chemist at Just, and I also just happened to become the ELN administrator when our company was pretty small and needed somebody to be the admin on our ELN. And so when our licensing was renewed, we kind of all talked about how much we didn't like our current ELN and how it wasn't really conducive to growth at Just, and we wanted to find something better. And so that kind of got put on me, and I formed this ELN team of wonderful people. These are just a couple of the people that are part of the ELN team, to try and find a better ELN. And so we evaluated a bunch of different options to try and find something that would work for us. Everything we evaluated was either too expensive, too floofy, didn't have enough, and didn't really provide us with what we wanted. And so then we decided to turn to LabKey, because we, store all our data with them and have so much going on in LabKey already that it just kind of made sense to put our ELN in a place where all of our data lived. And that kind of started off the conversation, and we started discussing like what we really wanted to have in an ELN. And one of the biggest things was making it easier for us to link in our data into our ELN, so we could stop duplicating all of our data in another ELN somewhere and storing that far away where nobody would ever look at it again, and kind of bring the data and ELN aspects together to make it, a little bit more of a fluid system at work. So ELNing is no longer such a hassle.
Abigail or Abby, would you like to talk about your role in this whole thing at all?
Sure. Yeah, I guess I was on the team of people who evaluated all the other ELNs that we were not fans of. And then I was also on the team that was working with LabKey to make the ELN. I feel like Tara just kept asking for volunteers, and she's my friend, so I kept volunteering. But it worked out really nicely, because I felt super comfortable just moving all of my ELNs to the new LabKey ELN with the rollout.
I was pretty late to the game. I was in the group that just kind of assessed the LabKey ELN. I didn't have to be part of any of the annoying parts before that. So it's been a pretty smooth process, I think.
Okay, thank you. Can you tell me a little bit about that process, the process of trying to get this adopted at Just, what that change management was like, any hurdles that you ran into, barriers to adoption, and maybe like what the state of that adoption is today?
Yeah, so for us, we decided we were moving forward with LabKey ELN. We started building that out. Our date for renewal was pretty set, or I guess our expiration date on our old ELN was pretty set in place. And so about a month and a half before that was rolling around, we just sent out an email to the whole company, and we're like, hey guys, this is no longer the ELN. Stop writing your ELNs in here. This is LabKey ELN. We're using this now. Send everybody the link to that, along with all of the LabKey documentation that they've created to kind of teach you how to use the ELN. Super easy, super user friendly, walks you step by step very simply through, how to create an ELN, how to reference things in your ELN, how to sign off an ELN, all which is already really user friendly and intuitive if you didn't even look at that. And right away, like within that day, people were already using the ELN, and our whole company has switched over to that. And I'd say in PD we have probably a little over 100 users now, and yeah, 300 plus ELNs in there already, and they're very easy to create. It didn't seem like we ran into any hurdles. I hosted kind of a meeting probably a week and a half after we rolled that out, just to kind of go over any questions people had, most of which the documentation answered, and gave people a little tutorial that was pretty similar to exactly what the documentation in LabKey provided. And yeah, since then there's really been no hiccups. Obviously there's people have suggestions for improvement or future features that we can work on, and obviously we're all ears for that, because we work with LabKey all the time, and they're here to help us make it better. So yeah, it's been great.
Yeah, I had an intern start maybe like a month after the rollout, and I gave her like zero direction. I was just like, it's in LabKey, you just click on the Biologics button, here's the notebook. And I assumed that we would like meet later and I'd walk her through it. And she just like wrote an ELN and submitted it to me. It was super easy, really easy for her to figure out, super user friendly.
Well, we're of course thrilled to hear that feedback. How has the change changed your day-to-day workflow? How does the new ELN kind of fit into that workflow, or how has it been affected?
The creation of just a lab notebook is so much easier now. I have a template for the small scale bioreactor group that it's really easy to reference our media batches. You just use the snipping tool to put in your graphs that you've made, which are also on LabKey Helper. And it's just, I mean, takes 30 minutes to do a lab notebook now instead of multiple hours of transferring information over to our old ELN system. And it never worked right. And it was a huge hassle. So now it's, we finish up an experiment and the next day my lab notebook is done and submitted and off to the next reviewer.
Yeah, I would agree with that. I also use templates for certain experiments that we do pretty frequently, and it just completely changed. I don't have to like rewrite everything because our old ELN had copy paste issues. And now when I'm writing stuff, it's instead of focusing on writing all the methods, I can just actually put my graphs in and be like, these are my conclusions, I'm done, move on.
Cool. What is the scope for a typical notebook for each of you? It might be different because you're in different teams, but number of samples, number of days of work, just in general, what's the scope for one of these for you?
Well, I can say for my group, since we use like the assay request system in LabKey very heavily for our day-to-day work, that is very, very much so where all of our data is stored. So writing ELNs for us really becomes like, here's my intro, here's my conclusion, here's all the reference samples that I used, here's the molecule that I'm looking at, and then like insert data from LabKey as a reference. And then now it's just like, oh, you want to go see the data? Just click the link. So instead of having to copy paste every single thing in there, it's really just very simple and very, very basic. I will say for other groups, I mean, we don't have a member of cell line development here, but they heavily use collaborative notebooks now. And so having be able to have 5 different people working on the same project, all harvesting different cells and working with the different cell lines, but it's all under one project, they can now collaborate on one notebook and have all of their information in one space instead of having, previously what would have been 5 ELNs separately, and then having to then work, to compile all that data together if they wanted to share that with the client. It's all in one space now. And so for them, they heavily use the day-to-day entry, which is a feature where you can, say on this day I did this, and then add a new day. And then on this day I did this. And so for them, it's very, day-to-day. They have all of their samples in there with the bulk referencing ability. You can, reference 20 plus samples really easily just by putting in all those numbers. And so it's not much more work than just adding one sample. So for them, it's been great.
Yeah, I mean, that sounds like a nice impact to efficiency and collaboration there. Abby or Abigail, have you? How does that impact on collaboration? What impact has this had on your ability to collaborate, and what does that look like?
I do a fair amount of like cell culture work, small scale cell culture work, and sometimes I'll have someone else take care of my cells for me on a certain day. And so if I'm not in that day and I need them to come take care of my cells, I can just add them as an author to my notebook, and they just sort of kind of write up like what they did that day. Or if they did like the initial culturing and then passed it off to me, they can sort of write what they did initially and then what I did once I took it. So it doesn't happen for me too much because a lot of my experiments are just my own, but also within my group, it's like something we all sat together and designed, and then like one of us did most of the main cell culturing and sampling, but everybody else was kind of involved. So it's nice to reference them as co-authors on that notebook, just so that we're all sort of like aware of what's happening and where the process is right now.
Yeah, I'll just echo that. The bioreactor group, the team lead does all of the paperwork. So there isn't a lot of collaboration with those, but we do, since the bioreactor group often works on the weekends, will passage cells for the cell line development group. And so they can just add us onto their notebook that they're keeping track of, every time they've passaged, and we just add in our information for a Saturday passage, and then now they have it for their own notebooks.
Yeah, I guess I work pretty heavily with one person in the cell line development group, sort of just helping her with her productions. And since I'm in media development, she'll add me as a co-author to a notebook where I maybe don't necessarily need to write that much, but she'll always ask me to make comments on the amino acid analysis of her cells that she normally wouldn't really put in the notebook because it's not really her thing. But since I'm on the notebook, it's pretty easy for me to just add that in.
Yeah, thank you. Let's see. So you've all mentioned use of templates. What would you say of all of the notebooks that you have to author? What portion of them come from templates, and what portion of them are novel or standalone?
100%.
100% templates.
Yeah,
Same.
all the bioreactor templates are entirely, I don't know, not.
Yeah. Yeah, I have like maybe when I'm actually developing a medium, I'll write sort of a different type of notebook, but since the rollout, I haven't had to. But I'm just assuming that when it comes to that, I'll just write a new template for that sort of experiment, and we'll use that from now on.
Yeah, I think across every group, the format of an ELN is kind of defined, like what you need to have in terms of like your introduction, your method, your conclusion. And so even if you are adding more text or, kind of deviating from the template, it's still pretty easy to just like start from the basic template and then add in more information if you want, but like at least the basic outline is there, and that takes away that extra step of having to do that.
Okay, and having all of that templatized, has that affected? Obviously it's going to speed up your authoring of these things, but has that affected your operations in other ways?
I think if anything, having a template and making that whole like creation of the ELN and just writing your ELN so much quicker is actually allowing us to put more information into the ELN, since so much of the basic information is there now. And the tedious aspect of writing an ELN, what used to take, hours across multiple days, now that it's so quick to just get the basics of your ELN, you kind of are there for longer thinking about like, oh, well, actually, I'll just add in this little blurb about like this particular filter that I used, or like how this was different from the last experiment, or and just more, verbiage around that. So if anything, our ELNs have more information than they did previously.
Yeah, I would agree with that. And I would say just the fact that it doesn't take hours anymore means that I can actually like get my other work done too. And I don't have to like stay home for an entire day writing the 5 ELNs that I'm not caught up on.
Okay, so that's a pretty large efficiency gain on the authoring side. How does the review and approval experience differ from your previous operations, and how has that affected your how quickly you can operate now?
It's been, well, for me, a lot easier. The previous system we used, it didn't have like reminder emails. So you would have to email the person, tell them a lab notebook was ready for them to review, and then hound them every week because they wouldn't review it. And especially now with the templates, specifically for our group, you write in your little bit about how the cells grew or the amino acid analysis, but really everything is the same over and over. So they just have to go in, maybe look at the 2 paragraphs that you've written that are different than the template, and hit or add whatever comments they want and send it back to you. And it's the reminders are what are just the best part for me, because then I don't have to constantly email people and remind them to look at it. they get bothered by the system itself, or every time they log in, like overdue, you need to look at this. So it's gone a lot faster, I think, in terms of getting things checked out and having the reviewer really like look at the information that you're providing instead of being overwhelmed by information. It's just like, okay, I need to just look at these couple paragraphs instead of the whole thing.
Yeah,
Okay.
our old ELN also didn't have a way for you to look at attachments without downloading them. And so that was like a huge pain, and it a lot of times people just like never looked at the attachments and assumed that you attached the right file. So it's I would say from an author standpoint, it's better because you know they're reviewing everything and not just the text.
Cool. Yeah, it's funny how impositions of extra energy can reduce quality and cause people to make assumptions like that. That's really interesting. You talked about comments between reviewers and authors. What are the ways in which you've made use of commenting within these notebooks, and how has that facilitated any change in your operations?
Well, I think that especially like the ability to be able to comment while you're making the ELN. And so if you have a collaborative ELN and you know somebody has on a different shift schedule than you, then it's really easy to say like, oh, I did this. And then if you're looking back at somebody else's like day and you're not really sure like, oh, is that actually what they did? Or maybe there's an error there, you can put a comment in right away. And so then when they're back in that ELN editing it before even the review process, they can see that comment and address that then, instead of having to, you know, wait for the reviewer and then maybe miss that and, you know, that you're missing information. And then even in the review process, you can also comment. And so in the review process, if you are reading through somebody's ELN and you're realizing like, oh, there's some weird stuff there, you can add comments in the same and then return it back to them with those comments. So I think it just all in all just, you know, makes it easier to talk to somebody through the ELN instead of having to previously, like you would email them and be like, hey, by the way, your ELN has some errors in it, so that you didn't have to send it back to them and resend it. Like it was a lot more of a process. And so the fact that it's just like where it needs to be, and then you get an email being like, somebody commented on your ELN. It's already like set up.
Yeah, I would second that. I'd say the previous method was like you message somebody in chat and then they'll say like, oh, sure, I'll fix that. And then they kind of forget about it because it was a chat and not like a real conversation. It's like something they say they'll do later, but with the reminders from LabKey, it helps a lot.
Very cool. I heard you talk about file attachments and review. What are some of the other scenarios where we're having to put lots of attachments on there is important, and has the LabKey ELN change? How else has LabKey ELN experience in that regard changed your operations?
For me personally, I used to attach all of my titer reports from Tara, and now I just reference them instead of attaching them. So that's really nice.
Meaning that the results data is in an assay or something.
Yeah.
Yep. I'll just put the assay request reference, and then somebody could click on it and it'll open that LabKey page. So I don't have to export it, save it, and then attach it to the ELN.
And on that similar note, with the assay function in LabKey, we're often putting like additional information, comments about, you know, like the data itself in there. And so now you don't have to just like duplicate that. So that if you just reference that, then you can click to that LabKey data, have all that information in there. And it's not a matter of like Abby having then to analyze my data and say the same summary that I already did. It's already there. So just like linking that in makes it that much quicker for sure.
Very cool. Okay. We have a couple of questions from audience members. I want to see if I can convey them reasonably to you. One of them I think will be straight to Abigail. There's a question about getting online time-dependent data from bioreactors into LabKey ELN. That's not something that we've talked about much previously. How does your group deal with that kind of data?
So time dependent. So we wait until, well, at least I do. I wait until the end of the experiment to put all my data in, because weekly we have our group meetings, whether it's with the client or just within our company, to go over that data. So the lab notebook, it's just kind of at the end, once I've compiled all the data, once I have all my amino acids and product quality back, then I'll make the lab notebook itself. We are looking at getting run sheets instead of using Excel run sheets like we are, maybe doing something in LabKey instead, a LabKey based run sheet. So that would give us real-time data, but that's kind of in the future and hasn't been, I don't think, even started yet.
Okay, Abigail. There is okay. There's a question in here about migration, but I think that will best be answered separately. There's also a specific question about comparisons to some other specific ELNs. I don't necessarily want this to be a compare and contrast, but maybe I could ask Tara or whomever is interested in generally summarizing, let's see, what you were looking for and why some of the other available ELNs didn't quite have what would serve Just-Bio's needs at the time.
Yeah, I think the biggest things we're evaluating when looking at other ELNs were cost and how that would scale with the growth of our company. We've grown quite a bit in the last year and a half alone, and so we've over doubled in size. And so for us picking a new ELN, we wanted to make sure that we were considering that cost and what that growth would look like. Additionally, the other big thing was how we could link in our data and how we could make that easier. And so every other option ELN that we looked at was a separate system. And so there were options to have your data then live in that system, but we weren't interested in moving our data over. We like LabKey and we want to be in LabKey, and we like the collaborativeness of the relationship we have with LabKey and what that allows us to do. And so everything then was kind of how can we integrate our data with this ELN and our data that lives in LabKey, and how can we make that connection? And a lot of these other ELNs had the option to do that. It would be building APIs to do that, but that is a task in itself. So we were just wondering, what's the easiest way we can just get our data in our ELNs and have that be linked? And that's where it just made sense to us to go with LabKey, just because all of our data lives there. And having the connection with LabKey that we do, we're constantly working with them to try and develop things to make our lives easier at Just. And so having our ELN there as well, would give us that opportunity to then continue to integrate into that ELN and continue to make it easier to put our data in there, and that relationship still exists. So we're constantly looking at ways to improve and what's going to be the next step and what we want to do and how we want to make things easier and improve our workflows. And so it just was a no-brainer to us for getting that piece.
Thanks, Tara. Let's see. So Tara kind of mentioned the fact that the data was already in LabKey for them, and so there's a question here about migrating existing data into LabKey. I think that's probably going to be best addressed separately. We'd love it if you'd get in touch with us on that. And for anybody who's interested in seeing the Biologics ELN, we'd be delighted to show it to you, of course. There is an additional question here about cell passage information. You talked about being able to stamp work onto a new day into the notebook. The question here is how do you record that cell passage information based on bi-cell measurements or others? Is it an ELN field, or is it in an assay or something else? Does one of you have an answer for that?
Most of the time, I just graph my passages if I think it's necessary for the point of the ELN. What we do most, if you're passaging in shake flasks, we count on the vi-cell, and we have our vi-cell pull directly into our network, which then is saving it in LabKey. So if you have the same sample ID for your passage, you can look up the sample ID and see all of the cell counts for that sample ID.
Super cool. Okay. I would like to mainly thank you, Tara, Abigail, and Abby, so much for your time today, and of course your efforts for getting this implemented. We're of course thrilled that you are finding success with it. I want to mention that currently the Biologics ELN is not standalone. Its value is dramatically enhanced by being built onto the structured data of LabKey Biologics. And we are exploring other scenarios for its use in our other products, and we would certainly love to hear from you, our audience, about your interest and your scenarios. So Tara, Abby, Abigail, thank you so much for your time. We really appreciate it.